Hello everyone, this is Ketish here. In this video, I'm going to go through the AQA Biology Paper 1 Foundation Tier from 2018. Now this is the biology paper from the separate science or triple science paper. This video is going to be a full paper walkthrough in which I'm trying to go through, or I will be going through, questions 1 to 6. This paper is actually out of 8 questions. Questions 7 and 8 are higher tier questions, and I have done separate videos for those question 1 and 2, and I will link that to the playlist at the end. So question 1 to question 1 to 6 are questions that appear only in the foundation tier. So let's have a look at question number 1. Now remember that you are going to be tested on 4 main topics in the biology paper 1. Topic 1 is on cell biology, topic 2 is on organization, topic 3 is on infection and response, and topic 4 is on bioenergetics. So question number 1 is on the topic of cell biology. This question is about the cell cycle. Chromosomes are copied during the cell cycle. Where are chromosomes found? That would be inside the nucleus. What is the name of a section of chromosome that controls a characteristic? That would be a gene. Figure 1 shows information about the cell cycle. So you've got cell growth, and then copying of chromosomes, and then mitosis. Mitosis is the cell division stage. So if I start here, and the first of all, the cell actually grows up to a certain point to its maximum size, and once it grows to its maximum size, then it starts copying down the chromosomes. It takes about half of its cell cycle. So copying of chromosomes is actually a long process, and it takes time. And then cell division takes about 30%. So if I look at in terms of percentage, that's 50%. I would say that's 25, right? Let's say 35% of the time, and this is 15% of the time. Which stage of the cell cycle in figure 1 takes the most time? That would be copying of chromosomes. During mitosis, cells need extra energy. Which cell structure provides most of this energy? That would be the mitochondria, where aerobic respiration takes place to release energy. So site of aerobic respiration. Ribosomes is where protein synthesis takes place. Site of protein synthesis. Cytoplasm is where chemical reactions take place. Site of metabolic or chemical reactions. And chromosomes don't actually take part in any function. They only have the code, which is actually just copied later on. The cell cycle in figure 1 takes 2 hours in total. The cell growth stage takes 45 minutes. So if the cell growth stage takes 45 minutes, calculate the time taken for mitosis. Okay, so let's have a look back at this. So cell growth is taking 45 minutes. They're saying this stage is taking 45 minutes. Now if you look carefully, they have divided into 1, 2, 3 parts. So each part is actually 45 divided by 3 is 15 minutes, right? So The question is asking the time taken for mitosis. That would be, right, and just 15 minutes here. 15 minutes, right? So 45 plus 15 will give me 60 minutes in total. This takes, copying of chromosomes takes 60 minutes, so it all adds up to 2 hours. So 15 minutes, we don't really need to show any working for this. And if you need to, you could say 60 minus 45 equals 15 minutes. Figure 2 shows some cells in different stages of the cell cycle. So you have cells labeled here A, B, C, D, and E. Okay, in which cell is not dividing by mitosis? Now, here you can see the chromosomes clearly. So if I use color, see these dark patterns, actually the chromosomes. So here the chromosomes are in different points. And this one also looks like you can see the nucleus really dark. That's actually right at the start of the cell division. Here in cell C, you don't actually see the chromosomes, right? So during mitosis, you won't actually see the chromosomes. Sorry, during normal cell growth or function, you won't see the chromosomes. So that is cell C. Cell E in figure 2 contains 8 chromosomes. Cell E divides by mitosis. How many chromosomes will each new cell contain? That would be still 8 cells, because remember, mitosis, right? Cell duplicates or copies itself into 2, right? So if it's a copy of the parent cell, it should still have the 8 chromosomes. Why is mitosis important in living organisms? To produce gametes. Now that is mainly by meiosis. Remember, there are 2 types of cell division. Mitosis is one, meiosis is the second, which produces gametes, which are basically sex cells, the egg and the sperm cell. To produce variation, not by mitosis. Mitosis copies itself to release energy, to repair tissues. Right? To produce variation, you still need meiosis as well. That brings us to the end of question number 1. Let's have a look at question number 2. Question number 2 is again on the topic of cell biology. Plants are made up of cells, tissues, and organs. Actually, this is not on cell biology, it's actually on organization. Question number 2, this part is actually starting on organization, because we're talking about cells, tissues, and organs. Draw one line from each level of organization to the correct plant. The organ is actually a leaf. A tissue, a root hair cell is simply a cell. A vacuole is a organelle. A xylem cell is simply a cell again. Spongy mesophyll is a tissue. You have 2 types of tissue in a leaf, the spongy mesophyll and the palisade layer actually have the epidermal cells as well. So there's different layers of cells in there. Now, figure 3 shows a plant cell drawn to scale. So you have the length equals 50 micrometers. Okay, 50 micrometers. Length of chloroplast, they have actually given you the arrow. where in a plant would the cell in figure 3 be found? You would find a palisade cell. It is a palisade cell, isn't it? They haven't told you what type of cell it is. It shows a plant cell drawn to scale. Now this cell is called a palisade cell. Well, how do I know that it is actually a palisade cell? Because it has lots of chloroplasts and it has this rectangular shape, right? So I'm looking at the shape and number of chloroplasts. Now let me quickly show you a diagram of the leaf structure. I think it's worthwhile having a quick look at it. So here is a diagram of a cross section of the leaf. So the top layer is the waxy cuticle, that's the shiny bit of waxy layer. And then upper epidermis, these are flat rectangular cells. Then you have the palisade mesophyll, which has got this rectangular column shape, and it has a lot of these chloroplasts. Now the spongy mesophyll also has chlorophyll or chloroplasts, but they are not actually in rectangular shape. They're actually of different shapes. And then you got the lower epidermis, which contains the guard cells and the stomata, and in the middle of it is actually the leaf vein, which contains the xylem and the phloem, which has two main parts. The xylem is actually responsible for carrying water, and the phloem is responsible for carrying dissolved substances. Right, so let's have a look back at the question again. Where in a plant would the cell in figure 3 be found? It's not in the epidermis, it is in the palisade mesophyll. Calculate the length of the chloroplast labeled in figure 3. Now, in order to do this, they're asking you to calculate in micrometers, so you don't have to do any conversion. It's only 2 marks. So they've already told you that this is 50 centimeters. So you will need a ruler to actually measure the length of this arrow. Right? Now, the length of that arrow is now, because I'm doing it digitally, it's a bit hard to, I'm trying to figure out how to actually explain. So if I measure the length of this is about 1 centimeter, right? And if I measure the entire length of this is about 8 centimeter. So if I divide 50 by 8, I should get the length of what 1 centimeter is, which is the length of the chloroplast. So 50 divided by 8 will give me 6.25 micrometers. Now, they're actually accepting a range between 6 to 6.3 micrometers. Right? Cells in plant roots do not photosynthesize. Give one reason why. Because they do not contain chloroplasts, right? Well, there's no point in having chloroplasts because it's not going to receive any light because it's the root cells, right? Or you could say chlorophyll. Right? Any two of those words will give you the mark. As a plant grows, new root hair cells are formed from unspecialized cells. Unspecialized cells. How does an unspecialized cell become a new root hair cell? It is by the process of what we call differentiation. Metabolism is about chemical reaction. Transpiration is losing water in plants, and transport is actually moving substances. Scientists can clone plants using tissue culture. Now, tissue culture, this part of the question is actually on the topic of cell biology. So you have the parent plant with a white flower. You take a scalpel, which is a knife, removing part of it. You remove a small section of the leaf and you put it in the growth medium, and it actually turns and starts growing roots, and each of those pieces of leaf that you put in the growth medium will start to grow into a different plant. Eventually, if you repot them, you would have the entire thing growing white flowers. The reason they're all white flowers is because it's literally coming from the same plant. So if I take small pieces from the leaf, let's say 6, and grow them in the growth medium, I would have 6 different plants, but they all came from the same parent plant. So they're all clones, right, of each other, because they'll have identical genes or chromosomes. Why might scientists want to clone plants? To create new species. If you want to create new species, you can't do it by cloning. To introduce variation, no. You can't introduce variation. To protect endangered plants from extinction, yes, that I would agree. Because if, let's say, you've got the very last plant, then you'd want to actually clone it so that you have at least more than one, so you can actually grow them and slowly start making a bigger number of plants. To reduce the disease resistance to plants, cloning doesn't actually reduce the disease resistance, it actually increases it. Or if you get one disease, if the parent plant gets the disease, and all the rest of the plants will actually get the same disease, right? So you're not going to make it resistant to disease. What is the advantage of cloning plants using tissue culture? No special equipment is needed. Maybe plants can be produced quickly. That I would agree. Plants are all different colors. Definitely not. The offspring are all genetically different. They are not genetically different. They are genetically identical. The main reason for producing plants by tissue culture is so that they can be produced quickly. You do need special equipments, at least the growth medium. 2.8. The growth medium in figure 4 helps the plants to grow. Name one substance in the growth medium. Now, the most common answer that students would actually say is nutrients, which is not right, because it's nothing specific. You need to name a substance. You can't give things like fertilizer as well, or plant food, because they're not actually naming a substance. You're just giving general names. So what would you actually have? Well, if plants are growing, the main thing that they would need is glucose. So you could say glucose, right? You could say hormones, right? Specifically auxins. And you could also say ions, minerals, such as magnesium. To make the chlorophyll, you need magnesium. In humans, for the blood, you need iron. Magnesium is needed in plants, and you also need nitrates, which is actually part of the fertilizer. You could say vitamins, or the simplest thing is water. You need water for it to grow. That brings us to the end of question number 2. Question number 3 is on the topic of organization. Figure 5 shows the human digestive system. So you've got 3 labels, and they're asking you to label organs A, B, and C. Now we can try and label all the rest of it as well. So let's label A first. Stomach. This is the liver. And this is the small intestine. what about the rest of them? This is the large intestine. Right. Now let me quickly go through the functions. Stomach is where the food is broken down, which mixed with the stomach acid, hydrochloric acid. Liver is mainly responsible for breaking down toxins in your food. The small intestine, where the glucose is mainly absorbed once it's been broken down. The large intestine is where water is actually absorbed. You've got the pancreas there. This bit is actually the pancreas, right? Which is responsible for producing insulin to control blood glucose level. This is the food pipe or esophagus. Obviously, that's your mouth with the salivary glands. And that's where the salivary glands are. Right. Complete the sentences. Choose the answers from the box. Digestion is the process of breaking down large food molecules into smaller molecules that are soluble. Right? Now, large food molecules are insoluble, so you want to break them down so that they are soluble. Enzymes help to break down food because they catalyze, which is basically to speed up chemical reactions without being used up. Remember, this it comes from chemistry. If the temperature of an enzyme gets too high, the enzyme is denatured. Which means it breaks down and it's not going to go back to its original shape. Protease is an enzyme. Protease breaks down protein. What is protein broken down into? That would be amino acids. Now, fatty acids, where do they come from? They actually come from fats and lipids. And glycerol is also coming from fats and by breaking down fats and lipids. And glucose is mainly coming from breaking down carbohydrates. Such as starch. When they're broken down, you will get glucose. So amino acid comes from proteins. Why is protein needed in the body? Now, you need protein for a couple of things, mainly for growth. You could also say for repair or replacement of cells or tissues or even organs. Right? Maybe not in humans, we can't replace the entire organ, but remember, a leaf is an organ. So if you pluck off a leaf from a plant, the plant can actually grow more leaves. Which organ in the human digestive system produces protease? Now, protein or protease, the enzyme is actually made in the stomach because protein is mainly digested in the stomach. Sorry, proteins are digested mainly by the enzyme protease in the stomach. So the stomach makes the protein protease. Sorry, enzyme protease. I'm getting it wrong. Question 3.6. Describe how you would test a sample of food to show it contains protein. Give the reason for any safety precaution that you would take. Now, before I continue, it's worthwhile going through different food tests because it's a required practical, right? So this question is part of a required practice. Although they're only asking for tests for protein, let me quickly go through the main food tests for 4 different things, and then we'll come and answer this. So this image or table actually shows the 4 main food tests that we actually do. Starch is the easiest one. We add iodine solution to it, and if there is starch, it will turn blue-black. You need to make sure that you write blue-black if there is starch. For protein, you add what is called Bioenergetics solution, which is actually slight light bluish solution. But if there is protein in the food, it will turn purple. For reducing sugar, right, like glucose, you would add what is called Bioenergetics solution, and then you would actually heat it in a water bath for a couple of minutes, and it will actually change from this blue solution into yellow, orange, or brick red. If the more glucose there is, the more red it actually becomes. And for lipids, you would add ethanol, which is basically alcohol, and you'll shake it, and it forms a cloudy suspension when you shake it, right? So the main thing is actually it turns cloudy or milky. So these are the 4 main food tests that you need to definitely know. There's a high chance that they can actually ask this in the 2026 exam paper. There you go. That's my first prediction. Right. So let's have a look at the model answer for this. Describe how you would test a sample of food to show it contains protein. So let's say I give you, let's think of something that contains protein. Definitely a piece of sausage, right? If I give you a piece of sausage, how would you test that it has protein? You would actually cut the piece of sausage into small pieces, and then you would put into a mortar and pestle and actually grind it into a thin paste. Then you would put the paste into a test tube with water and shake it. Once you shake it, it should release some of the proteins that was inside the food into the water. You could filter it. That way you will remove a whole lot of the bits and pieces from there, right? To remove all the unwanted pieces. Once you get a liquid which is coming from the crushed piece of sausage, then you would add Bioenergetics solution. Now, Bioenergetics solution is the name of the person who actually came up with the solution, so you don't need to know exactly what's actually inside the solution. What you're looking for is this positive result for it to go from blue color solution to purple. Right? You don't need to heat it or anything. You just simply mix it and shake it, and it'll actually turn purple. The more protein it has, the more purple it becomes. So here's a model answer. So the model answer is grind up the food, add Bioenergetics reagent to the food sample. So even if you didn't filter it, it'll still be fine. The protein solution or protein turns solution from blue to purple, or you could say lilac as well. Now that actually gives you the 3 marks, right? Give the reason for any safety precautions you would take. So that is important. You need to give any safety precaution that you would take. What would you do as a safety precaution? You would wear goggles to protect your eyes, right? Cleaning up any spills so that somebody else doesn't touch any liquid and wind up touching it. Why would you do that? Because Bioenergetics solution is an irritant, meaning that it itches because it is corrosive and it is poisonous. If too much of it is, a little bit of it is fine, but too much of it is poisonous. So you would want to drink it by mistake, right? So let's have a look at the next question. Complete the sentence. Obesity can be caused by a diet high in fat. Right? Complete the sentence again. Obesity is a risk factor for, now definitely not skin cancer, but is it type 1 or type 2 diabetes? Now obesity is a risk factor for type 2 diabetes. Now, how do you know this and how would you actually remember this? Because it's important that you need to know the difference between the, now type 1, think of it as the most common in a way, right? Type 1 is what you actually see, or we actually see in when we associate people with diabetes who need insulin injections. Right? When we talk about diabetes, we actually automatically think about, we don't think about people who are overweight. We just basically think about, oh, that means they might need insulin injection. That's type 1 diabetes. Type 2 diabetes, they actually don't need insulin. Well, there's no point in actually giving them insulin because insulin won't actually work to reduce the blood sugar level because their body has stopped responding to insulin of any sort, even if their own body actually makes it or from outside. So the only way they can actually reduce the type 2 diabetes is by actually reducing the amount of sugar in their diet, mainly by changing their lifestyle. That brings us to the end of question number 3. So let's have a look at question number 4 again, which is on organization. This question is about the circulatory system. Draw one line from each blood component to its function. So the blood is actually made up of a couple of things. Platelets, right? These are basically broken down. Old cells, right? Red blood cells, obviously you should know what red blood cells are. These are the donut-shaped or disc-shaped cells. The white blood cells, there are different types. There's a white blood cell which doesn't actually have a specific shape, which is called phagocytes, which are responsible for eating or engulfing bacteria. You also get these memory cells with this large circular nucleus. Right? So platelets, the function of them is to help the blood to clot. Red blood cells transports oxygen around the body. White blood cells destroys bacteria. Now, what actually then transports the urea and transports glucose? These two are basically dissolved in the blood plasma. What is blood plasma? That's basically the liquid part of the cell, which is mainly 90 to 95% water, and it has minerals, salts, and all other kinds of things dissolved in it. It's a slightly yellowish color liquid. Figure 6 shows cross sections of the 3 main types of blood vessel found in the human body. Each blood vessel is drawn to the scales shown. So you have A, and the scale shown is actually multiplied by 4, B is multiplied by 5, and C is actually multiplied by 7,500 times. So C is actually the smallest. And A and B are roughly about the same size, so A should be the largest. Okay, and it has elastic tissue and muscle tissue in both A and B. Now, C is actually just one cell thick, so this is actually a capillary which connects these two blood vessels. So what are these two blood vessels? Now, blood vessel B is an artery, and this is a vein. How would I know this? Arteries have this thick muscular walls and thick elastic walls. Veins have actually thin elastic walls, right? So they have thin, T-H-I-N, yeah, and thick walls. Why? Blood is actually going through in arteries at a high pressure, so you don't want it to burst, so you need strong walls. Whereas in the vein, blood is not actually under high pressure, it's just returning back to the heart, so blood is actually under low pressure. So which blood vessel has the smallest diameter? Right now, looking at the size, you should know the diameter is basically the length across this bit. If you don't pay attention, you would simply write it as B, because visually this has the smallest diameter. But the answer is actually C. Why? Because it's actually been multiplied by 7,500 times. So in reality, cell C, or this blood vessel C, is actually 7,500 times smaller. You wouldn't even be able to see it, right? Which blood vessel in figure 6 is an artery? That would be B. Reason, since it has thick, is it simply muscle? Yeah, muscle tissue, or you could say elastic tissue. Now you could also say that this hole, which is called the lumen, is a specific word to describe this hole, right? But now this is actually multiplied by 5 times, right? So this is actually the smallest. So you could say, right, has the smallest lumen as well. But I would simply stick with describing that it has a thick muscle or elastic tissue. Table 1 gives information about the blood flow in 2 people. So person A and B. A does not have coronary heart disease. Coronary heart disease is heart disease or disease of the heart, which is linked to blood vessels that is carrying blood to the heart. So if you look at, oh, that's not a good diagram of a heart, is it? Let me redraw that. Right. Okay. Now that's roughly about the shape of the heart, and you've got the blood vessels going in and out, right? And in there, I'm going to use a different color. If you look at a heart, it has blood vessels all around it. Why? Because the heart is actually a muscle, so it needs oxygen and glucose. So something has to carry glucose and oxygen for the heart itself, right? So the coronary arteries or veins are basically these arteries and veins, which blood vessels. So when you get heart attack, that's mainly because these blood vessels that carry oxygen to the heart is actually blocked, right? We are not talking about these blood vessels. It's these blood vessels that we are talking about. If they actually get blocked, that means your heart is not going to get enough oxygen, which means your heart muscles will start to die, and you wind up with what is called a heart attack. So going back to this, A does not have coronary heart disease. The blood flow through the coronary artery is 250 centimeter cube per minute. B has coronary heart disease. So because the blood vessels are blocked, that means the blood flow is actually slower and smaller. So it's 155. Calculate the difference in blood flow between person A and B. The difference is 250 minus 155. So that is about 95. Suggest why blood flowing through the coronary artery is lower in people with coronary heart disease. Right? Main reason is blood vessel B, which is basically the coronary arteries, right? Actually, let's call it coronary arteries. Put that. Arteries are blocked, right? That would give you the mark, but I'm going to continue. So they have narrower lumen compared to the person who doesn't have coronary heart disease, right? Arteries already have a small lumen, but people who have coronary heart disease will have a much narrower lumen. And you could say, what are they clogged with? With fat deposits. Calculate the volume of blood flowing through the coronary arteries of person A in 1 hour. Give your answer in decimeter cube. Now, again, this is actually a chemistry question. What does a decimeter cube mean? Decimeter cube is equal to 1,000 centimeter cube. Oh, and it's actually maths as well. So, how much does it carry in 1 hour? So, let's have a look at for person A. Person A does 250 centimeter cube in 1 minute. And that's per minute. So, because it's 1 hour, 250 multiplied by, which is basically 60 minutes. So 250 multiplied by 60 would give me 15,000 centimeter cube. Now remember, 1 decimeter cube is actually equal to 1,000 centimeter cube. So if I divide this by 1,000, it will give me 15 decimeter cube as the answer. 15. Coronary heart disease can be treated by inserting a stent using a coronary artery bypass graft or CABG. Table 2 gives information about each method. Now you need to know about stents, right? Or what is called a coronary bypass. Now let me explain the bypass. Let's say that's the heart, right? And you've got a blood vessel which is basically flowing through like this, and you've got a blockage like this. Now what would you do? You can't actually force it out because it will just go further down. You can't simply cut it and actually take out the fat or scrape off the fat and then repair it. The blood vessels are actually quite small, right? So if you want to do that, you'll have to do open heart surgery, which is actually quite hard. So what they do is actually they take a artery or vein, mainly an artery, from your leg muscle, from your thighs, right? From the back of your thighs, you can actually see blood vessels actually flowing through. They would actually cut a small section of it and then stitch it back up again. And they remove a small piece of artery and then join it like this, so that the blood actually goes through the side, bypassing this. This is why it's called a coronary artery bypass. Now, what is a stent? Now, stent if that is my blood vessel or artery, right? Now, and this is the fat deposits that is actually on the side, making it narrower. A stent is basically a thin metal mesh, right, which is basically made up of titanium, right? The reason they make it out of titanium is so that it doesn't actually react easily. What they do is actually insert this tiny mesh-like thing in there, right in the middle of it, and insert a small balloon to blow it. And because it's made up of metal, it actually pushes against the wall, making it bigger. So what you wind up is actually with a blood vessel which looks like this, right? And the stent literally pushes the fat to the side, and the stent actually opens it up by force, and you just leave it there, and till the person dies, the person will have a stent, right? So stent, the patient is awake during the procedure. A small cut is made into the skin. A wire mesh is inserted into the coronary artery via a blood vessel in the leg or arm or leg. CABG, the patient is not awake during the procedure. The chest is cut open. There you go, right? To do a coronary bypass, you need to cut open the chest. A section of blood vessel from the arm or leg is removed. It is used to create a small channel for the blood to bypass the blockage in the coronary artery. When procedure is recommended, why would you actually recommend these two different types of procedure? When only one blockage is present, you could actually use a stent. When multiple blockages are present, then you would need, so if it's in the blood vessel and you've got blockages there, there, and there, right, then you'd want to bypass it, all of them in one go. Time spent in hospital after procedure, 2 to 3 hours. If it's an open heart surgery, at least 7 days. Recovery time after procedure is about a week, and open heart surgery is 12 weeks. That's about 4 months. Risk of heart attack during the procedure is only 1%. Risk of heart attack during the procedure is 2%, is double it. Chance of failure within 1 year, right? The stent can actually get blocked up again. If you're not, once you have this diet, doesn't mean that you can just simply go on carrying out the same normal lifestyle. You'll have to reduce the amount of fat in your diet. So the chance of actually coming back is 40%. Whereas if you do open heart surgery, it's only 5%. It's actually much better, right? That's about 8 times better having an open heart surgery. But then again, the risk is double, right? And the recovery time is 12 times more, right? The reason there's a 40% chance of failure is because, if I go back to my diagram, right, you're not removing any of the fat, right? So the fat can actually seep in or actually start getting deposited again, right? Fat attracts more fat, right? It kind of sticks together. So all you need is a little bit, and the stent can actually move as well. So if people aren't careful, it can actually get dislodged out of the way as well. Okay, right. Question: give 2 advantages of using a stent instead of a CABGS. Now, 2 advantage of using a stent is no need, let me change my color, no need to stay in hospital for a long time after procedure, right? Or you can say fast recovery time, right? Or shorter recovery time. It's less invasive, right? You're not opening up the entire chest. You're only making a small cut, which is about a centimeter or two, right, on the side of your chest usually, right? If that person has too much fat around the chest, then they would actually go through the arm, right? So less invasive, right? Or you could say less risk of heart attack during procedure, right? If I'm trying to sell this, I would say there's a 50% less chance of having a heart failure, right? Or you'd say you only have 1% chance compared to the open heart surgery. Give 2 advantages of using CABGS, right? Now, CABGS or open heart surgery has a lower chance of failure, right? Now, lower chance of failure, they have actually said within 1 year, right? So I would actually follow that up. Lower chance of failure within 1 year. Maybe the chance of failure actually increases after some time, but they haven't actually given that. Or you could even say that there's only a 5% chance of failure as well. You can use the data, right? And the other advantage is you only need 1 operation to treat multiple blockages, right? You just need 1 operation to treat multiple blockages. I know personally people who have had actually 2 or 3 stents, right? If 2 or 3 stents, they might actually manage. If there's more in different places, they would actually do stents because it's much faster recovery and so on, right? That brings us to the end of question number 4. Let's have a look at question number 5. Is on the topic of infection and response, right? Aphids are small insects that carry pathogens. Figure 7 shows an aphid feeding on a plant stem. Now, if you have plants growing in your garden during the summertime, the aphids are very common. They actually this ash color insects, and they're quite small. And if you look closely, you'd actually see them. But looking from afar, you'd actually see as if the plant is just covered in ash, right? An aphid feeds by inserting its sharp mouthpiece into the stem of a plant. So it's very similar to a mosquito, how it actually feeds on us, right? So it has the sharp mouthpiece. After feeding, the mouthpiece of an aphid contains a high concentration of dissolved sugar. So the dissolved sugars, so in a plant, the sugars are actually in the phloem, which is usually towards the outside. The inner part is where the xylem is, where it contains water. So aphids literally put a hole through and literally draws up the sugar from the phloem, because there's always constantly sugar moving from the leaf towards the root, because the root always needs sugar. Which part of the plant are the aphid feeding from? They're feeding from the phloem. What is the process of transport what is the process that transports dissolved sugars around the plant? Not filtration, that's in chemistry. Respiration, now that's to produce energy. Translocation is correct. Translocation, trans means from one point to the location, is to different positions. Trans is actually to transport. Transpiration is referring to, it's the second time that we come across this word transpiration, is the movement of water through and out of a plant, right? Now, in a normal plant, right, it's constantly losing water through the leaves, right? Water goes in through the stem and then comes out of the leaf, right? It's basically like sweating. It's an important process to actually to make sure that the plant actually is cool enough as well, and you need water constantly being moved up, right, as long as there's water in the soil to keep the plant's shape as well. Plants infected with aphids have stunt growth. Explain one way the removal of dissolved sugars from the stem of the plant causes the stunt growth. Now, dissolved sugars is basically you're referring to glucose. That is actually the dissolved sugar, right? So if the aphids are actually feeding on the glucose, right, that means there is less glucose for respiration, right? Remember, respiration, you need to produce energy, or not produce, release energy through respiration, aerobic respiration, using glucose. Why do you need the energy? For all chemical reactions, you need energy, right? So the answer is there is less glucose, or you could say sugars, for respiration, so less energy is released, right? That these 2 points would actually give you the mark, right? I'm just going to add a little bit for growth to produce, actually, protein synthesis or cell division, right? All of these processes require energy. Most aphids do not have wings when they hatch. After several generations, some aphids hatch which have wings and can fly. Explain the advantage of the aphid of being able to fly. Now, if it can fly, then it can go from one plant to the other. Otherwise, it's stuck on the same plant, right? And if you're the 10th generation aphid, right, unluckily, if you don't have any wings, then we're going to compete with a whole lot of other aphids around you. So you need to be able to move to another plant so that you can actually survive, right? So explain the advantage. So aphids can fly to another plant, or even to another part of the plant, right? Otherwise, they'll have to keep walking and using energy as well. And these are tiny insects. So it's not as if they can walk the entire plant, roam around the plant looking for food without spending a lot of energy, right? Why would they need to do that? To get more food, right? Or maybe to find a mate as well. If they're looking to reproduce, then they need to find a mate, right? Why? Then there would be less competition. The leaves of some plants release oils onto their surface. Suggests how the production of oil on the surface of a leaf may protect the plant from aphids. Now, when plants actually are under attack, right? It's not just for insects. There are some trees in Africa which actually release these toxins, which actually makes the leaf bitter. So it actually stops elephant eating the leaves after some time, right? So this basically coming from the oil, right? How the production of oil on the surface of a leaf may protect the plant, right? The oil prevents the aphids. Right here, the oil on the surface is actually there to stop the aphids from actually attaching to itself to the leaf, right? Or you could say it is also a toxin as well. So prevents the aphid from attaching to the leaf, or you could say the oil could or may kill the aphids, or it just makes it unpleasant. It might be the smell as well. Figure 8 shows part of a rose plant. Give one adaptation shown in figure 8 that helps the rose plant to defend itself. That's mainly the most common thing. That's the thorns, right? So has thorns. Figure 9 shows a plant. Figure 9 shows a plan of a garden containing rose plants. The direction of wind. So you've got the plants in, there's 9 plants in rows, and they've got, okay, that's a key. Okay, so you've got 9 plants, and right in the middle is plant A. Surrounding it is B, C, D, and E. Plant A has a fungal disease, rose black spot, right? Rose black spot disease is actually a fungal disease that happens in plants. Which plant in figure 9 is the fungus likely to spread first? Now, fungus usually is spread by wind, right? So if the wind is actually blowing this direction, if the fungus is all over A, then it's going to get blown into C first, right? Give a reason. The plant is C, right? The fungal spores, these are basically like powdery spores are basically, how do I correctly say, they're basically like seeds, right, which new fungus can actually grow, but they're literally like powder, right? The fungal spores are blown in the direction of the wind, right? Marking scheme also says they accept it is the closest plant to A, but then again, looking at the diagram, they're all about the same distance from each other. So I would say it's mainly because of the direction of the wind flowing. Suggest one way the gardener could reduce the spread of rose black spot to the other plants in the garden. The best way is actually just cut off the plant that has the fungal spore and literally burn it, right, so that it doesn't actually infect others, right? Or you could, if you want to reduce the spread, right? Even before you actually get it, you don't want the plants next to each other very closely, right? So spread out the rose plants while planting, right? Or you could say remove and burn any infected plants, right? Or you could spray a fungicide. What's that? It's basically a chemical that would kill fungus. Question number 6 is on cell biology again. Earthworms are small animals that live in soil. Earthworms have no specialized gas exchange systems and absorb oxygen through their skin. What is the name of the process in which oxygen enters skin cells? That would be by diffusion. Remember, osmosis just to do with water, right? And active transport is again is to do with everything else except water, but it's against a gradient. Diffusion is down a gradient, whereas more to less, this is up a gradient or against a gradient. And respiration has nothing to do with transport here. Table 3 shows information about 4 skin cells of an earthworm. So A, B, C, and D outside percentage of oxygen. So there is more on the outside, more here again, more here again, and it's less, okay? Which cell has the smallest difference in percentage of oxygen between the outside and the inside of the cell? The smallest percentage, this is 1%. This is 4% difference. This is 2%. That's actually minus 4%. Right? The smallest percentage of oxygen between the outside and the inside is actually cell A. Which cell will oxygen move into the fastest? Now, oxygen is going to move from a high concentration to low concentration. So, into the fastest, that would be where the percentage is actually much higher. So, it's actually going to move much more in cell B into the cell, right? So, the reason I haven't chosen D is because it will actually move out of the cell, right? So, it'll move in the opposite direction. Earthworms have a large surface area to volume ratio. Suggest why a large surface area to volume ratio is an advantage for an earthworm. If it has a large surface area compared to the volume of it, then it can absorb. So it can absorb more oxygen in a given time because of the large surface area. Or you could say it increases the would not say the rate, increases gas exchange. Earthworms use enzymes to digest dead plants. Many plants contain fats or oils. Which type of enzyme would digest fats? That would be lipase. Protease break down proteins, lipase break down lipids, carbohydrates break down carbohydrates. Remember those. Earthworms move through the soil. This movement brings air to the soil. Dead plants decay faster in soil containing earthworms compared with soil containing no earthworms. Why? Now, if the earthworm is constantly moving in the soil, so if you pack a plant or soil, put a plant in the soil and actually just allow it to grow, remember the cells down here need to carry out respiration. For respiration, you would need glucose plus the oxygen. They can't carry out photosynthesis. So where are they going to get the oxygen from? The oxygen needs to come from the air in the soil. Now, if it's compact, that means there's going to be less oxygen. So what do the earthworms do? The earthworms basically digs these holes or tunnels. When they dig these holes or tunnels, that means more air can actually get into the root. That means more respiration can actually take place, right? For the plants to decay, dead plants to decay faster, you need aerobic respiration, right? That's carried out by decomposers like bacteria and fungi. So here's the model answer. Here's the answer for this. There is more oxygen in the soil with earthworms for more aerobic respiration of bacteria or fungi, or you could say decomposers. Each of those sentences will give you a mark. When earthworms reproduce, a sperm cell from one earthworm fuses with an egg cell from a different earthworm. Name the process when an egg cell and a sperm cell fuses. That is fertilization. Some types of earthworms reproduce by a process called fragmentation. In fragmentation, the earthworm separates into two or more parts. Each part goes into a new earthworm. So if you have an earthworm, if you cut it into two, they'll actually grow into two different earthworms. That's fragmentation. That's being a bit cruel, but it'll actually grow into two separate earthworms. What type of reproduction is fragmentation? Well, you started off with one earthworm and you cut it into two to grow into two. So that's asexual reproduction. Or you could say cloning. But the best answer is asexual reproduction, because it's a type of reproduction. You got sexual and asexual reproduction. Maybe you want to write the A right next to the S so that it doesn't look like as if they are two words. So asexual reproduction, right? This is sexual reproduction. When you get a sperm and an egg cell, that's sexual reproduction. That brings us to the end of question number 6. Now, question number 7, as I said at the start, is actually question number 1 from the higher tier paper, right, which is actually on the topic of infection and response. Is talking about salmonella symptoms of a healthy person. It's got a bar graph for you to interpret, right? And then again, results from a practical or investigation. It's out of 11 marks. And then the final question, question number 8, is on bioenergetics. Now, This is the only question in bioenergetics, it's linked to photosynthesis, right? And they're talking about question number 8 is actually in the higher tier question paper 2. So I will link 2 separate videos for each of these questions for the higher tier paper. Once you finish watching these 6 questions in this video, click on the playlist to watch question number 1 and 2 from the higher tier paper in order to complete these questions in the foundation paper. And again, it's actually about interpreting graphs, and there is a 6-mark question in here about exercise. And that's actually 20 marks, right? There's a total of 33 marks, or 31 marks, isn't it? So yeah, 31 marks just from the last 2 questions, right? That brings us to the end of the video. Thanks for watching. Hopefully you find this useful while you're revising your GCSE. I will link or share a video with you how to actually use these videos that I'm doing in order to help you to revise effectively pretty soon, hopefully. Thanks for watching. This is Ketish here. Bye for now.